thank you very much, Özlem, for great introduction. And so, as a disclosure, I'm a pathologist. I'm a renal pathologist. So I'm not an AI scientist. I'm not a computer scientist, but we do some work related to AI in my lab. And I will share those with you. So let's see if that works. These are my disclosures and learning objectives. Okay, so let's see. Yes, so as an introduction, as I said, I'm a renal pathologist, and also I'm a clinician scientist. Work at the University of Washington in Seattle. This is the old campus. This is, if you've heard or you've seen during the spring, the cherry flower blossom, that happens over here. And this is where my lab is located, Warren Magnuson Health Science Center, and we're in the 5th floor of that building. So it's a nice area, and you're more than welcome to visit my lab. If you happen to be in Seattle, please send me an email, and would love to give you a tour of what we do. So many people say that pathologists are not real doctors because we do not see real patients. We just see pieces of patients, like needle biopsies. But we deal with a lot of images at different magnifications. So this is a glomerulus at 400x, and we will look at that under electron microscope. We'll learn about some of the details at 5,000x. If we go higher at 12,000x, we'll learn more, and we'll learn more if we go higher as well. So we deal with images, and we deal with different kinds of images, different colors, different specialist stains, immunofluorescent microscope, electron microscopy images. We also deal with a lot of text. We have to put all of those together to arrive at the correct diagnosis and interpretation. So another way to look at these things is that if we digitize all those information, we actually deal with a lot of numbers. One can look at that this way as well. So on the research side, I'm very much interested into this famous quote from Lord Kelvin, that actually, I mean, for the science and also for the expressing our knowledge, it's very important that we have metrics. We can measure things, and if we can measure something, then you know that you know something about it, and then you can evaluate. You can make that better. You can make comparisons, and so on and so forth. And that's what got me into quantitative microscopy. And I got interested into histology or unbiased morphometry since I was in medical school, reading like books from material science and mathematics. So histology is basically a body of mathematical methods. Primarily, it's derived from stochastic geometry and deals with a lot of different sampling. Its goal is to understand three-dimensional structure in an unbiased way of complex-shaped objects through two-dimensional studies. And in pathology, as you know, similar to many of the radiology studies, we deal with two-dimensional most of the time, let's say that way, that we deal with two-dimensional images. So, for example, we're interested in measuring filtration length density in the glomerulus, or a very complex shaped membrane. What is the thickness, average thickness of that? What's the distribution of that? And that's a picture of me when I was still young and had hairs on, doing electron microscopy. And I remember that when I started my postdoc at Dr. Michael Mauer's lab at the University of Minnesota, it wasn't a century ago, it was 2001. And at that time, they were excited because they had recently acquired a digital camera connected to the electron microscope. So instead of stitching different images of a glomerulus, this is the glomerulus, the filtering unit of the kidney. So as you can see, these are like these spaces are are capillaries. The blood passes through them, and then filtration happens. So they used to, before digital camera, they were basically stitching images together, making montages, and then superimposing these grids and then doing the counting. Anyway, doing that manually or doing that basically using a computer software such as Adobe Photoshop was making things those methods were very powerful, and we were learning a lot about them. And I will give you some examples, and then how we are transforming those into automated way using AI. Now, this meeting is also about the lysosomal diseases or rare diseases. So I have to say something about my interest in lysosomal disease and Fabry disease as well. It was, in fact, Mike Maurer, who got me interested into the disease, came to me and said that, okay, I think that this is a very interesting disease, and there is a very cool question, and you may be able to figure it out. So Fabry disease, you guys are all expert. If there is one person who doesn't know about this disease here, it's caused by mutations in alpha-galactosidase A, leading in GLA gene, deficiency of alpha-galactosidase A, leading to accumulation of the substrate, which is primarily global triacylceramide, and that accumulation manifests itself as these multilamellar inclusions or GL3 inclusions in the cytoplasm. So if we look at different cell types, we see that pretty much many different, almost all cell types in the classic patients, they accumulate these GL3 inclusions, and some of them, they accumulate a lot of them, such as podocyte, which makes them vulnerable. Also, peritubular capillary endothelial cells, that we will hear about those a little later during my talk, they were regarded by FDA as like a center of attention for looking at GL3 inclusions and whether they are cleared or not following a given treatment, as at that time that treatment being agalsidase beta. And clearance of those cells from GL3 inclusions was regarded as efficacy of the drug. Okay, so the question that Mike Maurer asked me was about the podocytes, however, because at that time we knew that, okay, endothelial cells in peritubular capillary, they clear easily after enzyme replacement therapy. However, even longer term studies, even though that they were suggesting that some of the podocytes, they become partially clear, there was no statistical significance. So we were thinking about podocytes as resistant cells to enzyme replacement therapy. And why podocytes are important? Because these are post-mitotic cells that, they're very beautiful. They make these all these elaborate extensions that they interdigitate and make food processes. But the important thing is that these cells, they have very limited regeneration capacity. And if, this is the glomerulus, the same picture that I showed the electron microscopy of. If they lose about 15-20% of these cells, they scar down. And that's completely irreversible. So podocyte loss is cumulative. Mike asked me that, okay, how can we measure podocyte GL3 accumulation in a proper way? And so being interested in histology and measuring things in an unbiased way, basically we ended up modifying a technique that was called point sample intercept method that was based on the volume weighted, the star volume. So we made that less volume weighted and more appropriate for the kind of measurement that we were interested in. And that led, in fact, to a method that is applicable to many different cell types, including podocytes, not only podocytes, but also cardiomyocytes, endothelial cells, smooth muscle cells, and so on and so forth. We showed that, yes, podocytes after enzyme replacement therapy with agalsidase beta, they partially reduce their GL3 content, and that leads to shrinkage of the podocytes. And this is also partially associated with improvement of food process widening, which is a widely acceptable marker for podocyte injury. Same thing, we showed that following migalastat treatment, 6 months of migalastat treatment, we also have partial reduction in the GL3 accumulation inside the podocytes, and again, that improvement or clearance of the podocytes from GL3 is associated with improvement of food process widening, and also correlates with reduction of lyso-Gb3, which is another marker for severity of the disease. We also showed that, yes, accumulation of the GL3 in the podocytes is associated with podocyte loss. So, in fact, it better be predictable of progression, because the podocyte loss plays an important role in progression of many CKDs, including Fabry, as we know it. And we showed that these things are podocyte loss and accumulation of GL3 is correlated with age. So as we age, podocyte accumulation increases, and also podocyte loss increases as well. Okay, so all of these are saying that podocyte GL3 and injury markers, such as food process width, are biomarkers, are meaningful biomarkers for Fabry nephropathy. And when we are talking about the biomarkers, we get back to that quote from Lord Kelvin, that actually, we want the biomarker to be something measurable, objectively measurable. And the way that we were assessing podocyte injury and podocyte GL3 basically was quite quantifiable in an unbiased way. As I showed you, because of those properties that podocytes have, limited regeneration capacity, correlation of podocyte loss and injury with albuminuria and proteinuria, we can categorize that probably under several categories of biomarkers, such as prognostic biomarkers, and also predictive biomarkers, and also markers of drug efficacy. However, and this slide goes over that basically how we look at the sequence of events, that we have the GLA mutation, alpha-galactosidase deficiency, GL3 accumulation, that leads to increased accumulation of the inclusion, cellular injury, and eventually organ damage and organ failure. So we have to keep in mind that also we want a biomarker to be an early and sensitive marker of the injury, so that it can guide our treatment for basically preventing the progression before scarring and irreversible injury happens. And GL3 accumulation that starts very early during the fetal period, as well as food process widening, probably meet these criteria for being sensitive biomarkers as well. Among the different type of biomarkers that we can look at, general biomarkers that they show like function of the organ, and also imagings and different types. The cell-based and tissue-based biomarkers, including the GL3 accumulation in the podocytes, as well as food process widening, can be regarded, in fact, as specific biomarkers that specifically basically demonstrate injury to cells that are directly linked to the pathophysiology of the disease. So, importance of the early intervention in Fabry disease is very well known to all of us. This is a study that was done by and published by Dominique Germain, that showed in patients who have been on enzyme replacement therapy for a long term, those who have not been responsive in terms of preventing GFR loss in patients receiving agalycides beta. They were patients who had greater proteinuria, they were patients who were older, and they were patients who had more global glomerulosclerosis. All of these things are likely related again to the podocyte injury as well as aging. Okay. So is podocyte injury an early biomarker and a sensitive biomarker of Fabry nephropathy? Yes, we showed using the techniques that I showed you that in fact we have widening of the foot processes as a marker of podocyte injury happening early in life. These are in patients all below age 20, and widening of foot processes is progressive with age, similar to accumulation of the GL3 in the podocyte, which occurs early and is progressive with age as well. But how do we measure foot process width? Using the embryology techniques. So we typically study at least 3 glomeruli in a biopsy. We do systematic uniform random sampling. We study 50 to 100 images per glomeruli, and then those images that are obtained by systematic uniform random sampling, we superimpose these grids that are unbiased counting grids and basically measure the surface of the glomerular basement membrane as well as the slit diaphragms. These things are done by human or have been done by human. And the whole process was taking about 6 to 8 hours per biopsy. So this is a good biomarker. This is a sensitive biomarker. However, consider that 6 to 8 hours by an expert technician is such a long time, making this approach and this technology very limited. Even for clinical trials, it will be time-consuming and expensive. And obviously, what can help us to automate this process would be artificial intelligence. So how artificial intelligence helps us in that regard? Let's change the gear a little bit and go to the history of AI. AI is something that we had been dealing with for a long time. It's not unknown to us. So for last year, Nobel Prize was pretty much focused on related to the AI. The Nobel Prize of Physics, as you know, was awarded to Hopfield and Hinton for their work on developing protocols for Hopfield, for the Hopfield network, which was a model associated with memory and simple recurrent neural networking, and also to Hinton for building on Hopfield work and developing Boltzmann machine. That was an essential work that basically was related to artificial neural networking and made many things possible after that, as well as the Nobel Prize in Chemistry that was awarded to my esteemed colleague at the University of Washington, David Baker, for computational protein design, as well as Demis Hassabis and John Jumper for protein structure predictions. So David Baker, who has been working on how to predict protein structures and also designing the new proteins for years using artificial intelligence, was able to, for the first time, build entirely new kind of proteins. And that was to a large extent based on machine learning technique that Hassabis and Jumper had also created for predicting protein complex structures as well. So now we get into the idea that how about if AI scientists, we can have AI scientists. So basically, continuing what Dr. Alpern was saying, is AI capable of doing everything and create real science or not? So here, as you can see, the concept was put forward by several individuals that initially the AI does the literature search, comes up with the hypothesis. That hypothesis is also then evaluated by the AI. Experiments are designed, and then those experiments are being executed by the AI, and the data that is generated is also evaluated by AI in a circulatory fashion, and improvements are made, and eventually the AI also puts the manuscript together. The manuscript is reviewed by the AI, and if improvements are needed, those improvements lead to generation of new experiments. And these are examples of some of the papers that are generated and by AI scientists, and those papers are currently basically posted in the archives. Most of the people who have reviewed these papers, they think that AI-generated papers just do very small increments in the scientific knowledge at the moment, but there is great hope in that. Can we have a totally automated AI lab? So basically, the answer is that in this paper that was published in Nature in 2023 from A Lab, as you can see, the goal in the A Lab was to identifying novel air-stable compounds, and those compounds were predicted by Deep Lab initially, and also recipes were suggested by machine learning. Then those recipes were followed and were executed in a totally automated lab that is working by robots. And the products that were developed in the A Lab, they were analyzed automatically, and then the data was also reviewed. If the target was matched in terms of the efficiency and also having the properties that were predicted, they were noted. If not, new recipes were generated, and again, this going into the cycle of improvement and improvement. And it's very interesting to know that in 17 days of closed-loop operation, in the A Lab performed more than 300 experiments and successfully realized about 41 out of 58, which is 71% success. Excuse me. In basically making these novel organic crystalline solids. So with that, if AI can generate science, write papers, execute experiments fully, can AI also get a Nobel Prize? And that's the idea that was put forward by the Sony CTO, and as a challenge, Nobel Turing Challenge, that until 2050, probably we can have AI models that they do very valuable scientific work that is worthy of Nobel Prize by itself. And AI is now very much into politics as well. As you know, in Albany, we have now AI minister, that is actually like an assistant. It's in the cabinet. And as the Prime Minister of Albany announced, that is for tackling corruption issue in the Albany, and of course that's very good. They also announced that the AI minister is now pregnant with 83 digital assistants, that they will assist with the with the parliament as well, taking notes and assisting others. So these are all very, of course, attractive and interesting ideas, but at the same time, we have to be careful, and regulations are definitely needed, because everyone likes to be a friend of an AI minister, and those who have power need also to follow regulations as well. Okay, so this timeline shows that the progression that we have in AI in relation to pathology. So you see that the famous paper of Turing, and then the generation or coining the term of artificial intelligence, as well as deep learning, and so on and so forth. And actually, where pathology starts to be part of this story, maybe we can say that it starts with the papers that were published by Judith Prewitt. And in those papers, actually the scientific methodology is kind of very similar to what we are doing at the moment. So scanning the images, and also digitizing the data in terms of the intensity of the signal, and then interpreting that in terms of the chromosomes for chromosomal abnormalities, as well as after that the later works that were more focused on the identifying the abnormalities in relation to the cytology. The other important milestones in introduction of AI to pathology is related to starting using scanners, whole slide images, that creates this very large, like 20 to 40 gigabyte images that are used for pathologic diagnosis more routinely these days, but creates tons of data, and they have been used for pathology AI at the light microscopy level. And then at the end, what we have is that now AI tools in pathology are started to be regulated and approved by FDA, as well as other regulatory agencies, to be routinely used for diagnostic purposes, for example, for prostate cancer. There are many wonderful works and applications of AI in pathology. Most of those are in the field of light microscopy, however. The electron microscopy field that we are more focused on is less touched. So one of the, as an example of one of the wonderful works that have been done in Dr. Mahmoud's lab at Harvard and Brigham, is related, for example, the application of AI for predicting the origin of cancers of unknown primary. So in the pathology, when there is a metastasis, one of the big questions is always that, okay, I mean, where that metastasis is coming from, and that directly affects the treatment strategy. So in this work, Dr. Mahmoud's group, they provided whole slide images to a convolutional neural network. These images, as I said, they are very big, so they are broken down into tiles and patches. And then this convolutional neural network basically created vector-based and attention-based algorithms through deep learning to train which parts of the tissue is coming from a metastasis. So meaning that if we have, for example, this is, let's say, this is a lung tissue, if there is a tissue that is identified as an anomaly by the machine learning, whether that anomaly is coming from, I don't know, breast, or is coming from the bone, or is coming from somewhere else. And it was amazing that basically the model was shown to be performing very well across many different types of cancers with very high sensitivity and specificity, as well as what they did was that they asked the model to provide probabilities. So whenever there is uncertainty, we can basically come up with probabilities as probability number 1, probability number 2, and probability number 3 for the possible origins of the metastasis. And they demonstrated that the accuracy of the model in those 3 probabilities and options were also very, very good. Now let's go back to our work. We need to consider as one of the limitations of the AI, or as such as one of the limitations that we have to consider when we are doing any type of measurements, is that this first part, you saw that we did very meticulous sampling, in fact cannot be overlooked. So it's correct that we want to have a like fast analysis of the tissue or anything, but even if you are dealing with a very well-performing and robust AI model, if you do not provide an appropriate sampling, it can be garbage in, garbage out. So that's something to consider for any type of quantitative assessment. So at any rate, we aimed to generate deep learning algorithms for automated measurements of food process width, instead of superimposing those grids and a human sitting and measuring those elements for calculation of the food process width. So for that, we used a convolutional neural network based on encoders and decoders, and an architecture that is very similar to UNet and is called ForkNet. And with 2 masks in parallel. One mask is segmenting podocyte glomerular basement membrane interface, and the other mask is segmenting the filtration slits as dots. And then we were superimposing those 2 and then calculating the distance between the slits, which is equivalent to the food process width. In order to do this, we needed to do basically preparing a training data set, and for that we created a cloud-based segmentation utility that now many people in our group are using from different places, even from other countries. So, and then for the computer vision part, we had a membrane mask, created the membrane mask, we skeletonized the glomerular basement membrane, and then we cleaned up the data by doing some filtering, finding the open ends and cutting them, and also connecting them, and also cutting the branches, superimposing the slits on the PGBMI, and then eventually doing the calculations for the food process width. These are some of the output data that you can see. These are the raw images, glomerular basement membrane, slit diaphragm, and then superimposed images and the actual measurements in pixel. The performance of the model was pretty reasonable. So as you can see, the model training accuracy measured against the manual segmentation was about 65% on average, and also for the slits it was more on average, about 80–85% as well. So then we did we evaluated if the deep learning model that we generated was interchangeable with human measurements that at that time was gold standard. And for that, we compared deep learning and also expert technician, showed as ET here, measurements in different disease categories. So this is MCD's minimal change disease, which is an example of extensive food process effacement, and we have controls on the other end that the food process effacement is very minimal. And in the middle, we have patients, female patients with Fabry disease, male patients with Fabry disease, and patients with diabetic nephropathy. And as you can see, there was no significant difference between the human measurements and deep learning measurements across these different disease conditions. The second criteria for interchangeability is correlations. And as you can see, across these different categories as well as combined, we have very nice correlation between the methods as well. And also the third criteria for interchangeability is using Bland-Altman plots. And as you can see also across these different diseases, the Bland-Altman plot shows that most of the measurements fall between the limits of agreement between human as well as the machine learning. When we look at the different class comparisons, the agreement is a little bit less, and that's something to consider. Validation is a very important part of machine learning approaches. So meaning that if we overall across a wide spectrum of conditions, we have the method validated, still we need to validate that in each subcategory as well separately, especially if the model is going to be used for clinical purposes. But very interestingly, what we observed was that the deep learning measurements of food process width outperformed the human measurements in terms of correlations with relevant variables. So in Fabry nephropathy, as you can see, urine protein creatinine ratio and age correlated with both human measurements as well as deep learning measurements. But deep learning measurements showed correlations with podocyte number density, which is very important, but did not show significant correlation with human measurements. In the diabetic kidney disease, also deep learning measurements outperformed human measurements in terms of showing correlations with albumin-creatinine ratio, as well as known indices of structural parameters of diabetic kidney disease, including mesangial fractional volume and glomerular filtration density and podocyte volume as well. We have to always consider that in any experiment that we are doing, we need to overcome the noise of sampling. So basically the total variability in any kind of measurement or any kind of quantitative assessment is the summation of the error of the variance, which is noise, and the variation, which is coming from the biological phenomenon. Therefore, for that reason, it's very important to determine that how extensive the sampling and the sampling strategy should be to overcome the noise of sampling. For that, we performed bootstrapping with incremental sampling, and we showed that for the food process with measurements using deep learning, we need something between like 60 to 80 measurements. And after that, the averages that we obtain are quite stable. Also, now we have many more details about the parameters across different glomeruli and different biopsies, so can determine interglomerular food process variation and intraglomerular food process variation. In addition, we also, using these details, can have a look at the distribution of the measurements. And so these are like in the controls. As you can see, most of the food process with measurements are skewed towards the left. And as we enter into more severe podocyte injury, we have like the bucket more than 2,000 nanometer. We see that the foot process widening in that bucket increases substantially until we reach into minimal change disease, where we have extensive foot process effacement. So a concept of segmental foot process widening that is a common concept in renal pathology and nephrology, and is indicative of some level of podocyte injury, can be defined in a quantitative way using machine learning as well. So one of the main advantages of these type of approaches is that we can make them now available to many people. So now we have a website, we have this model, and we're adding our other models to our website, whereas people can take the images according to the instruction and upload the images into the website and obtain the data with no cost. So this, of course, for the clinical trials as well as for the clinical purposes, can be a very, very useful tool that leads to standardized ways of assessing structural biomarkers. Now we are working on using the more self-supervised or weakly supervised approaches for other quantitative measurements, including peritubular capillary endothelial GL3 inclusions. Here I'm showing an example of how the vision transformer and MAE that we developed is working. So in this approach, basically we covered different portions of the image, and then we trained the model to predict the original image. And as you can see, even when 85% of the image is masked, the prediction by the model is impressive, but does not give you details. For the details now, we are using another approach that I can talk about it if you want after the meeting or at World Symposium. So this is the way that we create the training data set using our segmentation utility. This is for the peritubular capillaries. This is the capillary. For the endothelial lining, as you can see, it's green. GL3 inclusions, which is kind of like orange, and the nuclei that are blue. And the model that we create is a self-supervised model with autoencoders, and then we basically define some downstream task for the model, and after we define that, we train, the model is trained for those specific tasks, here identifying, segmenting, and quantifying the endothelial cells and GL3 inclusions inside them. So these are some examples. This is the ground truth images of an endothelial cell in the peritubular capillary. As you can see, it's very thin, very fine cell, and this is for the GL3 inclusions. And here we have what the model predicted, and here is the superimposed. And once we have this data, we can calculate the fractional volume of the GL3 inclusions in the peritubular capillaries in a quantitative way. Again, we do bootstrapping. For every single parameter that we want to study, we do the same thing, and it shows that roughly about 60, 70 to 80 images can give us a pretty stable estimation of the fractional volume of GL3 in the endothelial cells. Applying the model to biopsies from patients with Fabry disease, we can see that the model can separate, in fact, patients with classical mutation and males on the left side from patients who are either females or they have later onset variants from controls very, very nicely. And we're talking about very small amounts of inclusions in these examples. Most recently, we are applying a foundation model. This is Biomed Parse that was created by Dr. Wang our collaborator at Computer Science and Engineering Department at the University of Washington. So it's very interesting because BioMed-Parse was trained on a wide range of images from like frozen sections to CT scans, MRIs, and so on and so forth. But the pathology that you are seeing here is primarily H&E, whole slide images. So this model had not seen any electron microscopy images during the training. However, this slide demonstrates the performance of the model that does, in fact, very well. If you just visually want to compare that, basically how the BioMed-Parse worked in annotating nuclei in different categories and segmentations. Compared to the prior models, also it was functioning really nicely. It's a kind of multimodal model as well because it has both visual and as well as language components. When we applied the model to images from peritubular capillaries, we see that surprisingly the model does really well. So I'm not showing you the data, and I invite you to our talk at World Symposium in next year. But I can tell you that our preliminary data are very promising, and in fact, it show that this new model shows that also there is a correlation between the global ceramide inclusions in the peritubular capillaries as well as lyso-Gb3, and also can detect treatment effect when there is partial clearance of GL3 in females with Fabry disease. We can apply all these methods and models into other modalities that are associated with images. Here I'm showing a picture of our work on the imaging flow cytometry, where we can identify and quantify podocytes with their GL3 inclusions inside the urine, and show very nice correlations between the GL3 in the podocytes with the proteinuria and albuminuria. So the future directions of computational pathology is all about multimodal machine learnings that can provide very valuable information that can be used not only for diagnostic but also for prognostic as prognostic markers, as well as also in the clinical trials and predicting that who will respond and who is not. And so, as also you saw in the example that Dr. Alpan showed, these models are coming into our daily practice in the pathology and medicine very soon, and are currently perhaps are here as well. I mean, the model that I showed you in Dr. Mahmoud's lab is currently being used at Brigham Women Hospital. So we are also working on developing multimodal models for renal pathology, where all different modalities from electron microscopy to light microscopy to immunofluorescence to clinical information are all integrated together, and actually can basically provide something like an AI assistant for renal pathology. So these tools hopefully will be available in near future. There are some roadblocks and challenges preventing AI applications. Of course, the regulatory issues and ethical principles are very important. Validation is very key, of course, in the applications of the AI. Quality of data, as I mentioned about garbage in, garbage out, we have to consider that. Putting regulations together and clinical adoption of the AI is also something that is actively people are thinking about. And there is some resistance as well. I mean, there are some worries about the application of the AI from the pathology sides too. So AI has limitations. Of course, some limitations that are related to the cost, limitations that are associated with consumption of the electricity, consumptions of the water for the cooling system, creating wastage and so on and so forth, as well as political things that are into development as well as expansion of AI applications. At the end, for the sake of time, I pass through these quickly. So there has tremendous growth in the development of novel AI approaches in the pathology in the last few years, and currently actually AI is being actively used and will be more used in the near future in pathology as well as other principles. AI can improve diagnostic workflows, can eliminate human errors and increase inter-observer reproducibility and make prognostic predictions. As I showed you, they've revolutionized our ability for image analysis and can be very powerful in developing robust and standardized structural biomarkers that can be widely available. So for example, I showed you our website, and other applications can also become available to people either for free or at a very limited cost. And AI can increase accessibility of highly specialized assay for rare diseases as well as for clinical trials and in clinical grounds as well. So at the end, I would like to thank all the students in my lab, David Smerkus, and also Andy. They're in the computer science department, and they did very significant work. Other students played a very important role in training of the AI models as well, and also the other scientists in my lab who provided support. Many of the images were provided by Michael Maurer, and also specimens from his lab. Dr. Shang Wang from the computer science department played an important part in the Biomed Parse project. And also I would like to thank all collaborators who provided biopsies to us, as well as sponsors of our studies. Thank you.